Good day, kids. Mr. Mechnick here with a podcast on meiosis and a little history of the Mechnick family. So, here's my beautiful family. And if we look at some of the characteristics that we all possess, you can definitely see some identifiable characteristics and some uniqueness that each of us contain. Obviously, some of the characteristics that my children received are from Dad, and some of the characteristics are from Mom. But none of my three children look alike, and part of the reason is because of meiosis. So when we look at meiosis, we're going to talk about the rules of the sharing game. And as you can see, the cuteness gene that my three children have obviously came from, yeah, you got it, my beautiful wife. So when we look at meiosis, we're going to talk about the process. We're also going to focus on what happens during each of the steps to create the uniquenesses that all of us have. So first off, meiosis is a process that divides cells and produces gametes. So another name for a gamete are sex cells. So when we do this, it allows for the recombination of genes from one organism and another. And so this can be simply done by reducing the chromosome number in half. So in humans, for instance, we start with a full set of chromosomes in a diploid cell. So a diploid simply means that you have two full sets of chromosomes, one set from the mom and one set from the dad. So when meiosis occurs, it reduces that chromosome number in half to produce 4 haploid cells. So simply by making sex cells and allowing these sex cells to recombine to make the organism, it allows for some genetic variability. So if we look at the stages of meiosis, we'll see that it goes through the same exact steps that we learned about when we studied mitosis. So it goes through prophase, metaphase, anaphase, and a telophase, but then it goes through the entire division process a second time, allowing for the reduction of the chromosomes to occur. So when this happens, it takes all of this genetic information found in the parent chromosomes and divides them twice to produce only half the amount in the daughter cells. So these daughter cells are also called the gametes or the sex cells. So the other way that meiosis creates a lot of variation and differences, and again, this is why siblings often don't look alike and why they might show some similar characteristics as their parent. But what happens is during meiosis, there's the scrambling of specific forms of genes that each gamete receives. And this happens because of independent assortment and crossing over. So when organisms go through and make these gamete cells, we don't know which chromosomes are going to split in one direction or another. But what we do know is that when these chromosomes separate from each other, each gamete will receive a different set of instructions in the form of DNA. And as a result of independent assortment and this process of crossing over, it creates a lot of genetic diversity, which allows for organisms to evolve over long periods of time. Independent assortment is the process of random segregation and assortment of chromosomes during the formation of gametes. So if we look at this sample cell, there are a total of 6 chromosomes, or 3 homologous pairs: chromosome pair number 1, chromosome pair number 2, and chromosome pair number 3. So it gives us a diploid number of 6. So when meiosis happens, it takes all of the maternal chromosomes, the ones from the mother, and the paternal chromosomes from the father, and pairs them up during prophase. But as it goes through the first meiotic division, it will separate these chromosomes from each other. After the second meiotic division, the chromosomes will independently assort and separate. So if we think about all of the different possible combinations of gametes that we can end up with, since we're dealing with 3 pairs of chromosomes, we can see that there are combinations that can produce a red maternal chromosome with a brown maternal chromosome and a blue maternal chromosome. That's one gametic combination. Another combination would be this paternal chromosome and this paternal chromosome and this one. So that's another combination. But if we went through all of the possible combinations of how these gametes are formed, there's a total of 8 gametic combinations of just these 6 chromosomes. So this mechanism of independent assortment can alone create lots of variation. We can calculate how many different possible combinations of gametes there are simply by taking 2 to the nth power. And n is the number of unique chromosomes, which in this case is 3 pairs of chromosomes. So if we do 2 to the third, it gives us 8 different combinations. If we think of a human, in a human we have 46 chromosomes. That's 23 pairs. So we know that those pairs of chromosomes, if we look at how they can assort independently of one another, we basically have to do 2 to the 23rd, which gives us 8,388,608 different gametic combinations of gametes. So every single time a male produces a sperm cell, there's 8,388,608 different combinations. That's a lot of different combinations alone. So this mechanism is one way that can create lots of biodiversity. The second mechanism that can even create more genetic diversity is what's known as crossing over. So if we think of a chromosome that contains 2 sister chromatids, when these pair together in prophase with the homologous pair, you form what's called a tetrad. So a tetrad is referring to 4 chromatids that are paired up. This is chromosome pair number 1, let's say. During crossing over, there is going to be an exchange of DNA from one chromosome. In this case, the paternal chromosome will share information with the maternal chromosome. They cross over genetic information. As a result, this new chromatid has information from the father's genome. This paternal chromosome, the blue one, now has maternal DNA on it as well. So if this happens to be the chromosome that gets passed along to their children, it will have characteristics of both mom and dad. So crossing over, again, can increase the likelihood of certain alleles being passed along from parent to offspring. So with those two processes, independent assortment and crossing over, we get a lot of genetic differences. And as a result, siblings tend to not look identical. So when we look at meiosis in a single chromosome pair, we start with the diploid number of 2. So if that's my diploid number, we should automatically know how many gametes or how many chromosomes will be in the gametes at the end. And in this case, we're reducing the number in half, so we end up with only 1 chromosome. So during the entire process, chromosomes pair together, they align in the middle in metaphase, they separate in anaphase, and in telophase, they will eventually form 2 new cells. In telophase, these cells are not going to be identical because they've crossed over and they have separated independently from one another. The second division will occur and you end up with 4 unique gamete cells. So meiosis occurs in 2 divisions, and during this step of prophase 1, this is where crossing over happens. So make sure you note that prophase 1 is when crossing over happens. And some of the other terminology that I've been using, paternal simply means from the father, maternal means from the mother. So when you pass along certain characteristics that I passed along to my beautiful little daughter, Michaela, you can see that she definitely has received some of her genes from her father. So what makes a boy or a girl? Well, in the male, when the male goes through spermatogenesis to produce sex cells, one of the gametes could have an X chromosome, another chromosome could contain a Y chromosome. These are your sex chromosomes. The X and the Y will determine which gender the child will be. So in boys, the father contains the Y chromosome and there's a 50-50 chance of them passing along either the X or the Y to their children. Whereas the female contains two X chromosomes. She will always pass along an X to the son and to their daughter. So the male is what determines the gender of the offspring. The process of making an egg in a female is called oogenesis, and this is the pathway. An oogonium cell, which is a diploid cell, two full sets of chromosomes, goes through meiosis 1, meiosis 2 to produce four gamete cells. Three of the cells are considered polar bodies and are not going to be reproductive cells. They will be degraded and broken down and not used. The egg, on the other hand, becomes that reproductive cell that the females will produce once a month during ovulation. So again, this process is meiosis in females that produce the egg cell. So once a month, women will go through and make this egg. Whereas males, on the other hand, will go through a process called spermatogenesis. And during spermatogenesis, males start with a spermagonium cell. They will go through the first meiotic division, divide these cells into two. So we start with a diploid value of 4. It divides once. It divides a second time to produce spermatids with 2 chromosomes each. So when meiosis happens in males, they go through this many, many times a day. And so every single time they go through this process, there are 4 sperm produced from one original cell. And as a result, in a given day, a male might produce somewhere around 250 million spermatids. Whew! That's quite a few. So if we think about the importance of making sperm and making these eggs, so when there's this miraculous event, when the sperm and the egg join together, they will create a zygote cell. The diploid zygote contains both sets of chromosomes from the mom and from the dad to create their new little zygote cell, which is going to produce the new offspring. So as a result, meiosis allows for uniqueness, genetic diversity, and it's all the result of sexual reproduction that occurs in many, many living organisms. And there you have it, kids. That's meiosis. Thanks for listening. Please bring any notes and questions to class tomorrow so we can do a little follow-up activity to make sure you understand the importance and the process of meiosis.